tuning in to Planetterrian Daily, episode 62, coming to you on May 13th, 2026. Let's get into today's research and health developments. Research has decoded the two enzymes plants use to build mitraphylline, a rare twisted molecule with anti-cancer properties. Research has showed that senescent zombie cells surviving chemotherapy rely on the protein GPX4 to avoid iron-triggered death. Removing this protection caused the cells to self-destruct, shrinking tumors and extending survival in mouse models. The work points to a vulnerability that appears after initial treatment rather than during the first round of damage. Most people picture every damaged cell either repairing itself or dying quickly, yet a third fate is far more common than once assumed. Right now, as you listen, a small fraction of your cells have entered a permanent arrest, where they neither divide nor clear out, instead secreting inflammatory signals that affect neighboring tissue. These senescent cells accumulate steadily with age and after certain treatments such as chemotherapy. Their persistence helps explain why tissue function declines even when the original injury has long passed. One practical takeaway is that future therapies may focus less on preventing all cellular damage and more on clearing the lingering senescent population once it forms. From the persistence of damaged cells, we turn to another long-standing production barrier that has now been solved at the enzymatic level. Scientists at UBC Okanagan identified the two enzymes that assemble mitraphyline's unusual structure in plants, such as kratom and cat's claw. The work removes a long-standing barrier to producing the compound sustainably instead of harvesting trace amounts from tropical species. The discovery that two specific enzymes create mitraphyline's distinctive twisted ring system opens a route to microbial or plant cell production of the compound. Because the molecule occurs naturally in only trace quantities, scalable synthesis could finally allow researchers to test its activity against multiple cancer types in meaningful quantities. The same enzymatic logic may apply to other rare plant alkaloids, whose structures have remained inaccessible. If production barriers fall, mitraphylline could move from a botanical curiosity to a practical lead compound. While plant biochemistry offers new molecules, viruses continue to exploit host machinery in ways that basic virology is now mapping in detail. University of Maryland researchers mapped how enteroviruses, including polio and common cold strains, recruit both viral and host proteins to copy their RNA. The detailed mechanism functions like a molecular switch that decides between replication and protein production. The finding identifies a shared vulnerability across multiple enterovirus strains. On a different note, two randomized trials examined switching from injectable GLP-1 agonists to either an oral agent or a microbiome-targeted supplement. Both approaches showed potential for preserving weight loss achieved during initial treatment. The results point toward practical maintenance approaches beyond indefinite injections. Damage to microscopic blood vessels precedes overt heart and kidney disease, yet current imaging methods are impractical for routine screening. A new blood-based assay detects these early microvascular changes with potential for wider clinical use. Early detection is also advancing in neurology, where a second blood test for Alzheimer's has now received European clearance. Roche received CE mark clearance for an additional blood assay that can both confirm and rule out Alzheimer's disease. The test builds on an earlier Eli Lilly partnered assay already in use. Faster analysis of tissue samples themselves is the next efficiency gain. Charles River Laboratories launched a digital pathology platform that uses artificial intelligence to accelerate slide analysis and reporting. The system targets routine workflows in toxicology and safety studies. It claims at least a 1-week reduction in turnaround time. While tools accelerate data, Stories remain one of the most effective ways to convey complex science. The movie Project Hail Mary demonstrates how complex topics in astrophysics and linguistics can be conveyed without sacrificing accuracy or entertainment value. Well-crafted films reach audiences that traditional science communication often misses. New findings on cellular persistence and viral machinery show how basic biology keeps delivering concrete targets for both treatment and prevention. Next time, we'll be watching for updates on how the GPX4 pathway performs in larger animal models and human cell systems. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.